Well, welcome back to our Bio180 lecture series. Today, we are going to be continuing our exploration of Unit 5, cell processes. Previously, we've gone over the material related to membrane transport across a membrane. And now we'll be starting our next series, which is cell communication. We're going to start out with cell signaling, just kind of an overview today, and then we'll get into actual mechanisms of some of the receptors and transduction pathways that are important in cell signaling. So, we'll start with an overview of cellular communication, and then we'll talk about the different types of cell signals, and then we'll have a preview of some of what we'll be going over later with receptors and transduction pathways. So, for multicelled organisms, there can be, some scientists estimate, as many as 65 trillion cells in the body. And all of those cells, in order to form a whole organism, have to be able to communicate with each other and coordinate their activities. And that's accomplished through the process of cell signaling. So as an overview of cell signaling, you are in all cases going to have some kind of cell, and this will be our signaling cell, or our sending cell, and it is going to release, the general mechanism for this is some kind of signal molecule. So we'll put in kind of a vesicle, and you have some kind of signal molecule that's going to be released. So we might call this in some cases you might hear it called the primary messenger. And that signal molecule is going then to travel to some kind of recipient cell. So we'll have the recipient cell over here. And there's different ways that it will travel, and this will be our different signaling types. So cell signal types. So how this signal molecule reaches out to the receiving cell varies. And then on the receiving cell, we're going to have some kind of receptor present, and the idea is that the signal molecule will then bind to that receptor, and then we're going to get some kind of movement. That signal then through the receptor is able to move across the plasma membrane. And we have the next step of what's called transduction. And then the transduction pathways will eventually lead to some kind of response in the receiving cell. So we're going to have the signal sent, we have reception, transduction, and then response. And that is the basic paradigm of signaling, that you're going to have some kind of signal that's received, transduced inside the cell, and then you'll have some kind of response from the cell. We'll go ahead and then talk about different cell signal types. And for this, the best way to go over this is with a table. So we're going to talk about 5 different cell signal types. And so we'll put this type up here. And so we've got 5 different types. The first type is going to be direct contact. Then we'll have cell-to-cell interaction. We'll have a type of signaling called paracrine signaling. endocrine, and then finally we'll talk about synaptic or neuronal signals. So as we talk about these different types of signals, some of the categories or features, characteristics that we want to look at is the specificity of the signal, the range of the signal, or the distance of the signal. We'll try and describe a little bit about what happens. Give you any special keywords, and then maybe an example of where that might take place inside the cell. And then we'll give maybe some kind of analogy to human communication and how that works. So, in the case of direct contact, let's start with our description right here. You are going to have our signaling cell, in this case, is right next to our recipient cell. So here's our donor, and here's our recipient. And they're really right next to each other. And then they're connected to each other. The cytosol of one is connected to each other through what's called a gap junction. So it uses gap junctions. And so in this case, there's not even a receiver. You'll get some kind of signal produced right here, and it will flow through those gap junctions and enter into the recipient cell. And a lot of times, that signal will even be a currency, so some kind of ion or membrane potential. So an ion flux. Okay? And the place where this is most commonly seen is in heart contraction. And so some of the muscle cells of your heart are actually connected to each other through these gap junctions right here. And as you initiate a contraction in one cell, the membrane potential, or the change in the membrane potential, flows through these gap junctions, causing the different heart cells to contract in series with each other. And so over here, the specificity of these direct contacts is very close and very specific. So in this case, the donor cell is only signaling a single recipient cell. So it's very specific, and its range is very short distance, meaning they're right up next to each other and touching each other. In our next example, so now let's do cell interaction. We'll do this one in purple. And in cell-cell interaction, you are going to have our donor cell, and the donor cell will have some kind of ligand on it. So the signal is actually going to be attached to this cell, and then you'll have another cell that has a receptor, and so the two will kind of handshake together. And so you'll have a cell-cell interaction. This happens most commonly, for example, in the immune system with T4 cells. So in this case, you'll have some kind of display receptor, and the immune cell will actually come along and see if it can match that cell. And if it recognizes that cell with its receptors and something that's being made in there, then it will actually end up killing that cell. So you don't want these cells to recognize something. But that's the type of interaction they're using. The ligand isn't released at all. It's held on the surface of the membrane. And so these two are in contact, direct contact with each other. And so that's cell-cell interaction. So over here, again, this is very specific. We are targeting a single cell to communicate with, and also very short range. Okay. In our next type, it's paracrine signaling. And so in paracrine signaling, you are going to have a single cell, and it does release a cell signal. So there's the signal that it releases. But this signal is short in distance. So it's released into, so signal is released into the surrounding extracellular matrix. Surrounding region. And it's only going to affect cells that are in that region around it. So as an example of paracrine signaling, we're here in blue with paracrine signaling. An example of paracrine signaling is, again, in your immune system. So whenever you get cellular damage, so for example, if you were to get a bee sting or something, or you cut your fingers, those signals, you'll release histamine. This isn't the only paracrine example, but that's an example of a paracrine signal. And so you release histamine in that localized area, and that causes other cells to respond. So for example, the histamine might cause the blood vessels near you to become leaky so that you can get additional blood flow, and particularly white blood cells flowing into that wound site. It can also recruit then white blood cells to come into that area and acts as an attractant for them to allow healing to begin. And so that's an example then of paracrine signaling. The specificity, this is more general. So that signal is sent out. It's not targeting, the histamine doesn't target a specific cell, but it's just released generally, and will target many different types of cells as long as they have a receptor for them. And so it's a general type of signal. And the range is short. We would call this a localized range, meaning it's just around that surrounding area of the cell. Our last two are going to be kind of more long range. And so we will, for endocrine signals, here our signaling cell is going to release some kind of hormone or signal molecule. So there's our signal. And then that signal is going to enter into some kind of blood vessel or lymph vessel or duct of some kind. So we'll have a blood vessel here. And it has to enter into that blood vessel and then be carried to the rest of the body. Some examples of endocrine signaling are things like adrenaline, which is actually norepinephrine. And so adrenaline is released from certain cells, enters into the blood vessels, then travels throughout the body, and it will cause a variety of reactions of cell responses because it will hit a lot of different cell types. And so over here, endocrine signals then are very general. Again, any cell that has a receptor for it will receive it, and they are long range or distant, because they can really contact anywhere in the body. Our last signal type is synaptic signals. And this is one of the common ones. This is also known as neuronal signaling. And in this type of signaling, I think we have room over here, we are going to have a cell, so we'll have some kind of nerve cell. And that nerve cell will have a long axon process to it, this long, thin appendage right here. And we are actually going to propagate some kind of nerve signal along that axon. And then our recipient cell is going to be right next to it, and there is a small space between those called the synapse. And so the recipient cell does release some kind of hormone or signal molecule from the end of that axon, and it travels just a short distance and is received by that recipient cell just across that axon or that synapse. But most of the signal is traveling along the axon. And that allows synaptic signals to be very specific. They usually target just one or two cells. And then on the range, we usually tend to think of these as distant. You might also think, well, the synapse, the space that the hormone is traveling across, is very short. And that's true, the synapse is very short. But the axons can extend quite a bit of distance. So some axons, the longest axon that you have is a cell. You have a nerve cell that rests its body at the base of your spine. And then its axon extends all the way down your leg and ends at your big toe. And so that's a pretty long axon. So that's why we call this long distance signaling. But you can also think of it as a short distance signaling across that synapse. As you look at kind of these different types of interactions, there are kind of human analogies you can make to them as far as looking at specificity and then range. So for example, under direct contact, what might be the human communication analogy to that? Well, this might be something like maybe a kiss, where you're right up next to each other and interacting very closely. On the other hand, a direct contact might be something like a handshake or a high five. A paracrine signal is going to be where you are, again, it's a fairly localized signal, but it's going out to anyone. Notice that this is a very specific one. You can only handshake with one person, or hopefully kiss one person. But when you talk about a paracrine signal, we are going to, this is kind of if you stand in your backyard and just shout, or if in your room and you shout. So shout in a crowded room or backyard or anything, and you just shout out. Okay? So again, it's general. That shout can be heard by anyone in the area that has ears to hear it. But it's not going very far. It's fairly limited in how far that signal travels. When you talk about endocrine signaling, though, this would be more like a radio broadcast, a radio or TV broadcast. So that signal is being sent out over a very long distance, so much farther than you could shout, and it's general. Anyone that has a radio or a TV to receive that signal, that has the right receptor, is going to receive it, so it's general. And then finally, our last one was synaptic signaling. And so synaptic signaling is going to be more like a phone call. Okay? And a phone call is a good analogy for this. The distance, say when you pick your phone up and put it to your ear, that distance between the phone and your ear represents sort of the synapse of that. And then this is very specific. Again, you call only a single person or maybe one or two at a time. The synapse is short, but the distance can be quite long between them. And so that's just a table that shows some of these different cell signal types and gives you a little bit of information about them. So now, if we come back over here, and well, we've talked about this area, so we've been talking about the cell signal types, the types of signals that are released from a signaling cell and then might be picked up from a recipient cell. Over the next couple days, we're going to spend time looking specifically at different types of receptor and transduction mechanisms. And so, for example, receptors, we are going to learn 4 different types of receptors and how they operate, how you activate those receptors and how you inactivate those receptors. So we'll look at receptors. And we're going to get into details on the mechanisms and how they work. So we're going to learn about 4 different receptors. We are going to learn about intracellular receptors. We are going to learn about ligand-gated ion channels. We are going to learn about tyrosine kinase receptors. And we're going to learn about a new one, an important class called G protein coupled receptors. And as you watch the next video of these different receptors, it's going to be important to understand not only how each one of these receptors gets activated, but then how it gets deactivated as well. And then these receptors are the initiating point for receiving a cell signal. And then their job is then to pass along that signal to the next step in the pathway, which is going to be our transduction pathways. And so that'll be our final video of this series. So today, not too long of a video. We're going to go ahead and cut this off, and we'll be looking forward to seeing you next time or in class. So we'll catch you then. Bye-bye.